the photosynthesis chapter. This first slide, this first diagram that you see, summarizes all the steps in photosynthesis. I'm going to show you how to walk through all the steps of photosynthesis, showing you how the flow of the electrons move through the system. And if you can explain all these steps the same way I explain it, you should be sitting quite well, as long as you comprehend and understand what it is that you're saying. But before we get into that, let's zoom out and talk about parts of leaves and so on. So leaves are the major locations of photosynthesis. Chloroplasts is the organelle in which all of photosynthesis takes place. Chloroplasts are found mainly in cells of the mesophyll, the interior tissue of the leaf. Each mesophyll cell contains 30 to 40 chloroplasts. And CO2 enters and O2 exits the leaf through microscopic pores called stomata. Keep that word straight. Stomata and stroma are two different words. Keep them straight. One way you can keep them straight is that stroma has an R. You drink fluids through a straw. It has an R. Stroma has an R. So stroma is the fluid found within the chloroplast. So in this one example, the structure in the very middle is one chloroplast. And inside you have a whole bunch of thylakoids. A stack of thylakoids is called a granum. So you see granum is a stack of thylakoids. And there are a whole bunch of thylakoids inside of one chloroplast. And wherever you don't have thylakoids, but you just have space, you have a fluid called stroma. So that's stroma. However, up at the top there, you have a cross-section of a leaf. And leaves have pores, and so those pores are called stomata, letting CO2 in and letting oxygen out. On hot, very, very, very hot days, those stomata will close up to prevent water from evaporating out of the cell. But that also can be dangerous and toxic because oxygen then cannot get out, and that can be poisonous, that can be toxic to the plant. The plant wants to get rid of oxygen and wants CO2 to get in to undergo photosynthesis. So you have stomata, we have chloroplasts, we have thylakoids. These again are those green sacs in the chloroplast. And finally we have chlorophyll, which is the pigment which gives leaves their green color. The chlorophyll is what absorbs the light energy and transfers them to electrons. Be sure to be very familiar with the reaction of photosynthesis. 6 CO2, 12 H2O, and light energy is converted into 1 glucose molecule, 6 oxygens, and 6 H2O. The overall chemical change during photosynthesis is really the reverse of that of cellular respiration. If you just move that arrow the other way, so it's pointing to the left, you really have cellular respiration, where you start with oxygen and you start with glucose, you consume it, and you get out ATP, which replaces light energy. You get out ATP and water and carbon dioxide. And so this little diagram just sort of shows where everything moves, but you don't particularly have to memorize which atom ends up being which atom in the product. But it's just a nice little diagram. This is another nice diagram that summarizes the reactants and the products of each of the cycles. Notice coming into the thylakoids, you have water and light, and those are the two of the three things you need for any plant to grow. Imagine yourself, you got a plant in your, sitting in the window of your house. You need three things. You need sunlight, you need water, and it needs carbon dioxide. Well, the two of those three reactants are required for what we call light-dependent reactions. The light-dependent reactions are those on the left-hand side. The Calvin cycle is what we call the light-independent reactions. Light-independent meaning you don't need light for the Calvin cycle to move forward. It can occur at any time. It can occur during the day, and it can occur at night. Light independent and light dependent, which is on the right-hand side. So if you have light and water as reactants, you also have a couple more reactants. Notice you have NADP plus and ADP and inorganic phosphate. Those are also technically reactants. Without those, you can never get the products out of the light reactants, which are ATP and NADPH. ATP comes from ADP plus inorganic phosphate, and NADPH comes from NADP plus and hydrogen. Notice that some of the products of the light reactions are actually the reactants of the Calvin cycle. Do you see that? And so, if we only talk about the light-dependent reactions, or the light reactions, we're only going halfway through the story. The other half of the story then is the Calvin cycle, because the final product that we get, that we are really looking for out of photosynthesis, is actually glucose, which is the main product coming out of the Calvin cycle, which is that arrow pointing down. Overall, you notice that there are several products. We have oxygen, ATP, NADPH, and glucose coming out. And the reactants would be light coming in, water coming in, carbon dioxide going into the Calvin cycle, plus you have the products, the byproducts of the Calvin cycle, which are NADP plus and ADP and inorganic phosphate. When we talk about photosynthesis, we talk about the electromagnetic spectrum and visible light, which is a part of that electromagnetic spectrum, a very, very small area. And notice the wavelengths from 380 nanometers to 750. And it's the chlorophyll, or the pigments, that absorbs certain wavelengths and reflects others. Well, this diagram here shows that that chlorophyll, the green pigment of the leaves, tend to absorb lots of the reds and lots of the purples. And if it absorbs those but doesn't absorb the rest, that means that the rest are reflected. And if it reflects and doesn't get absorbed, it reflects and enters your eye, and that's the color that you see. Therefore, you see green. And so this graph here shows the absorbance of wavelengths. Look at chlorophyll A up at the top there. It's absorbing lots of those purples, the blues and purples, 400, 450, 500, and but it the graph goes down all the way until it reaches the 650, 700 range back up. So it absorbs the purples and the reds, but it doesn't absorb the greens, and that's because it reflects the greens, and that's why you see leaves as green, if that makes sense. So you have chlorophyll A. You also have chlorophyll B, which is another pigment that's structurally a little bit different than chlorophyll A, but close. And you also have carotenoids, which are another accessory pigment that absorbs slightly different. But overall, you notice that none of them really absorb those greens, and that's why they reflect the green, and that's why you see green when you look at leaves. Okay, so that's what's going on with that electromagnetic spectrum. That's what I want you to know. When now let's go back to our photosynthesis diagram and talk about exactly what's happening step by step. If you notice, there are actually two sets of photosystems. The two green boxes are photosystem 2 and then photosystem 1. Why in that order? It's in order of discovery. The photosystem 1 was discovered first, and electron transport chain 1 was discovered first, and later on it was found that there's actually two more that actually occur even before those two. And so you have, in the exact order, you have photosystem 2, then electron transport chain 2, then you have photosystem 1, then you have electron transport chain 1. Okay, let's follow the flow of electrons through this entire photosynthesis system. First of all, we have to energize electrons. We do that through the sun. But where first do we get these electrons from? You take water and you split it. That's why water is at the bottom left-hand side. You take water, you split it into its constituents. You have oxygen, O2, you have hydrogen, but you also get a few electrons out. Those electrons enter photosystem 2 and sit at those special pair of chlorophyll molecules. And now the electron is ready to receive energy, to become energized. And so you see at the top left, light energy. The photons come in, and it's the chlorophyll. The function of the chlorophyll is actually to absorb that energy, and the energy is bounced from chlorophyll molecule to chlorophyll molecule, and they actually give it and transfer that energy to the electrons, and that's the function of chlorophyll. The function of chlorophyll is chlorophyll can absorb energy from the light, from light energy, and transfer it at the same time, transfer it to electrons, and so that's how you energize electrons. If you take a look at the gray arrow all the way through that whole system, the, the direction of the arrow symbolizes how much energy the electrons have at that particular time. And so the up arrow in photosystem 2 is depicting how the electron is gaining energy. So you notice in the first green box, you have an up arrow. And then the blue set of proteins there, the electron transport chain 2 is a protein complex. Proteins put together. Each of those blue blobs are a protein, altogether a protein complex. But you notice that the arrow eventually moves down. The gray arrow moves down, which means that the energy is depleted out of the electron. And then look at photosystem 1. Once again, the arrow points up. Energy is once again put back into the electron, energized, and and once again, energy is depleted out of the electron in electron transport chain 1. Now let's go back one more time, start from the beginning, and let's talk about the movement of and energizing these electrons. Where do we get the electrons from? We get them from breaking up water, which is why we need water. Which is why you need water when you water your plant in your room next to the window. That's the use of the water. That's what we actually need, are those electrons. Those electrons are put into photosystem 2, where chlorophyll absorbs light energy and transfers that energy into the electrons, and those electrons are energized and eventually move into what we call the primary electron acceptor, which is that little purple box called the primary electron acceptor. And from there, the electrons then can move to the electron transport chain number 2. What is the exact function of the electron transport chain 2? It's a hydrogen pump. It uses the energy in those electrons to pump hydrogens against concentration gradient from the stroma, if you remember that word, stroma, from the watery portion of the chloroplast into inside the thylakoid space. And so you see how there's lots and lots of hydrogens inside the thylakoid, but not so many in the stroma. That's because of electron transport chain 2, pumping hydrogens into the thylakoids. It takes energy to pump anything against concentration gradient. Those hydrogens then are going to want to escape, going down the concentration gradient. It doesn't take any energy to go down the concentration gradient. It takes energy to pump against concentration gradient. And where do we get that energy from? From those energized electrons that just came from the photosystem 2. Well, why would we spend all kinds of energy? Why would the plant spend all kinds of energy pumping hydrogens into the thylakoid space? Well, the answer is because now those hydrogens are going to leave the thylakoid, but they're going to leave through a very specific protein called ATP synthase. ATP, through the kinetic energy of those hydrogens as they swoop through the ATP synthase, it binds, it glues, it synthesizes ADP plus inorganic phosphate. It glues those two molecules and makes ATP, our primary energy currency, ATP. What does hydrogen do again? It's through the kinetic energy, through the motion of these hydrogens flowing through from the thylakoid to the stroma through the ATP synthase. it will allow the binding of ADP and inorganic phosphate to make ATP. This is one of the products of the light-dependent reactions, but that's only half the story. Those products will eventually become the reactants of the Calvin cycle. So, if I were to ask a question, does the electron transport chain create ATP? In a way, yes, but not directly. Very, very directly. If we're very specific, the electron transport chain 2 is just a hydrogen pump. And then the hydrogens will naturally want to diffuse back out to equalize the amount of hydrogens, the space between the hydrogens, and so they diffuse through and move through this ATP synthase. And we're going to see in the cellular respiration chapter how this ATP synthase molecule is actually quite complex with rotors and things that turn and move. And but for now, they move through, and through their kinetic energy, they glue ADP and P together, making ATP. But now, way back up at the electron transport chain, we have a depleted electron. This depleted electron goes back into, now, it enters photosystem 1. And there we have more light energy being absorbed by the chlorophyll and transferring the energy to the electron once again. Electrons are energized and move to the primary electron acceptor, and there the electron moves to now the electron transport chain 1. What is the function of electron transport chain 1? It makes NADPH through the binding of NADP plus and hydrogen and that electron. All together makes NADPH, and it's NADPH and ATP that are two very important reactants for the Calvin cycle. Keep those. This might seem pretty easy, but as soon as we get into the cellular respiration, we're going to have even more of these terms that sound very, very close. For example, NADH. NADH is also an energy-carrying molecule, but it doesn't have that little p. So once we start talking about cellular respiration, and we're talking about photosynthesis, it's very easy to get all these molecules mixed up. So there's a way to remember that NADPH belongs to photosynthesis, and that's because the first letter of photosynthesis is a P, and NADPH has the P, is the one with the P, and so you can remember that NADPH belongs to photosynthesis, but NADH is the molecule in cellular respiration. We'll also find ATP in both photosynthesis and cellular respiration. Okay, now we are halfway through the story of photosynthesis. Now what occurs during the Calvin cycle? Again, the Calvin cycle is the light-independent reactions. It doesn't require light in order to move forward. Now, in a way, you could think of it, yes, it kind of does, because it can't move forward without ATP and NADPH, and we did require light to make those products. So in a way, yeah, but if we look at it just the Calvin cycle, technically speaking, it doesn't require light to move forward, but it does need three reactants: CO2, ATP, and NADPH. And if you notice, the products of the Calvin cycle, two of them at least, NADP plus and ADP, we actually do need as reactants for the light reactions. And so you see there's a cycle back and forth, back and forth, back and forth. ADP, ATP, ADP, ATP, NADP plus and NADP ATP. So you see that there's a cycle. The products of one end up being the reactants of the other, and vice versa. This diagram here is showing the exact same thing that we just went through, just slightly with slightly different pictures. And the reaction centers of the photosystems can also be called P680 and P700 because of the primary wavelength that they absorb, if you remember, from 450 all the way up to 700. So you might see those, but otherwise in the electron transport chain, we're just going to use those two electron transport chain. Don't worry about all the separate proteins like PQ and cytochrome complex and PC, and don't worry about those. And so this whole thing, you can look at this diagram, sort of shows, allows you to see the flow of electrons in another way. You take a photon, the photon energizes the electron, and so the electron jumps up, and then the electron transport chain 1 uses those electrons, uses the energy in those electrons to create a hydrogen high concentration gradient, a proton gradient inside the thylakoid, and eventually makes ATP. And then once again, the photons from photosystem 1 once again energizes electrons, and so you see the electrons jump back up, and then those electrons are used up once again to make NADPH. Now let's talk about the Kelvin cycle. The Kelvin cycle is also called the Kelvin cycle regenerates its starting material after molecules enter and leave the cycle. The cycle builds sugar from smaller molecules using ATP and reducing power of electrons carried by NADPH. Some of the terms that I want you to be familiar with is G3P. Also, Rubisco is the enzyme that starts the entire process. And then RUBP is ribulose biphosphate. We'll take a look at these. Here we go. All right, so take a look at the very first molecule that enters the cycle. Count up the number of carbons. There's 1, 2, 3, 4, 5, 6. There's 6 carbons. Those 6 gray balls are 6 carbons. Well, 6, and there's 3 of those molecules, so 6 times 3 is 18. So we have 18 carbons. And it gets converted into 3-phosphoglycerate. Count up the molecules. It has it breaks it up into 3 carbon molecules, but now there's 6 of them, so 6 times 3 is 18. It's the same number of carbon atoms. And that gets broken up into 1,3-biphosphoglycerate. Count up the molecule, count up the carbons, 18. Count up this is converted into G3P. Count up the number of carbons. These are 3 carbon molecules times 6 of them, 18. So you see that the number of carbons never changes. Everything is accounted for. But now, one of those G3Ps exits the Kelvin cycle. And therefore, as you continue through the cycle, you now only have 5 G3Ps. That's only 15. But 15 plus 3, it makes 18. So everything is accounted for. Continue on, and there's your ribulose biphosphate. Count up the number of carbons. 1, 2, 3, 4, 5. 5 times 3 is 15. And there you have it. Everything is accounted for. Well, you ask yourself, why is the first molecule then 18? Well, the answer is because um, you have carbon dioxide. You have 3 carbon dioxides up at the top. So 15 plus 3 gives you 18. The first molecule um, is 6 carbons large, and so everything is accounted for. The reason why I took you through this whole cycle that way by counting the carbons is to show you that everything is accountable, and everything should make sense as far as carbons coming in and carbons leaving. Now, notice in the cycle there is ATP that enters to make certain molecules. Notice ATP, the arrow going in, and ADP leaving. And notice also on the right hand side NADPH entering and NADP plus leaving. On the left hand side of the cycle, there's some more ATP that enters and some leaves. This is exactly where we use ATP as the reactants and NADPH as the reactants, and where the products are coming out, ADP and NADP plus and inorganic phosphate. I'm not going to ask you to memorize how many of these ATPs enter and where they enter. I just want you to remember that ATP is a major reactant of the Calvin cycle, and NADPH is also a major reactant of the Calvin cycle, and the byproducts are, of course, their sister products, which are ADP and NADP+. Now, if we go through this cycle only one time, we only get a half a glucose molecule. So actually, the Calvin cycle must proceed twice in order to make one full glucose molecule. And so let's go back again. So I hope this diagram makes a little bit more sense now. You see you have the products, the reactants of the Calvin cycle, which are ATP and NADPH and CO2. And through cycling twice, we'll get out one full glucose molecule and ADP and NADP+. All right, the rest of this is not talking about the flow of electrons through photosynthesis, but a few extra tidbits that I'd like you to know. Dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants close their pores, their stomata, which conserves water but also limits photosynthesis because you can't get CO2 in and you can't get rid of O2, which is toxic to the plant, if those pores are closed. And so the plant can undergo another cycle, another process called photorespiration. It's not favored, but it does allow, and it doesn't make glucose, but it does keep the plant alive. It's not favored, it's not preferred, but it does keep the plant alive. Most plants in the world are C3 plants. This is what we just talked. This is the cycles that we just talked about. Initial fixation of CO2 through the enzyme rubisco forms a 3-carbon compound called G3P, or 3-phosphoglycerate. In photorespiration, however, not photosynthesis and not cellular respiration, but photorespiration, when those stomata close, rubisco actually uses up that poison, that toxic oxygen. It actually adds O2 instead of CO2 in the Calvin cycle, producing a 2-carbon compound. Photorespiration actually consumes O2. We usually don't see this in plants, right? O2 is a product and leaves the plants and CO2 enters the plants. But in photorespiration, O2 is consumed and organic fuel and the plant releases CO2 without producing ATP or sugar. It keeps the plant alive, but it's not actively making glucose, which is needed for the plant to survive. Photorespiration limits damaging products of light reactions that build up in the absence of the Calvin cycle. In many plants, photorespiration is a problem because on a hot day, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. The Calvin cycle that we talked about, with adding up the accounting for all the carbons, we also call carbon fixation, converting molecules from one to another, to another, to another, all the way through. We also call carbon fixation. There are also plants called C4 plants. These aren't as common as C3 plants, but they also exist. These plants minimize the cost of photorespiration by incorporating CO2 into 4 carbon compounds. There are 2 distinct types of cells in the leaves of C4 plants, the bundle sheath cells and the mesophyll cells. I'm not going to require you to know specifically what they do or even how they do it, but I just want you to be aware that there are plants called C4 plants. They're not as common, but they are especially useful in hot, dry climates. C4 plants as well as cam plants. Cam plants, some plants including succulents use crustacean acid metabolism to fix carbon. Cam plants open their stomata at night, incorporating CO2 into organic acids, and then the stomata close during the day and CO2 is released from organic acids and used in the Calvin cycle. So here's a comparison between the C4 plant and the cam plant. C4 plants and cam plants are found in hot, dry climates. But most of the plants that we have in the world are C3, which live in moderate temperatures. We're talking your oak trees and your pine trees and your maple trees and your grasses and your bushes and your flowers. All these types of plants. Your typical plants are C3 plants, but your cacti and so on that you'll find in the desert, those are your C4 and your cam plants that are especially good at surviving in hot temperatures. That is the basics of photosynthesis, and I hope that you will be able to explain the flow of electrons and what occurs inside the chloroplast in the same way. And you will be sitting very, very well if you can do it in the same way, explaining to someone and of course understanding what it is that you're seeing. This concludes the photosynthesis chapter.